Alright guys, welcome to another exciting edition of Random Noises Miss Mystery's Cat Makes, also known as your lesson on cell parts. Apologies in advance, I left my graphic tablet at school, so there will be no little drawings on this lesson. So, all cells do a number of things. One of the things they do is they all maintain homeostasis, they all make proteins, and then they all pass their traits on to daughter cells. And these are important because this is related to the types of structures every cell has. So, every cell on the planet is made up of some combination of organelles. Organelles are the subunits of cells, and they're the basic units of structure and function. So depending on the organelles that a cell has, that's going to determine the functions of that cell. That's going to determine what that cell is capable of. So depending on which organelles you have in a cell, you might have a cell that is going to be producing a lot of proteins. You might have a cell that is going to use a lot of energy and therefore have a lot of mitochondria. And this is a common theme you'll see on AP test questions that ask about organelles. They'll ask you to draw conclusions about, based on the function of the cell, what types of organelles would it have in it, or vice versa. So, how do we study cells? Well, we study living things with different instruments. So there are some things that we're able to see with the naked eye. You're familiar with all of those because you all have eyeballs. However, there are some things that we need to magnify, and we need a light microscope to see those. So we can see a lot of bacteria. We can see sometimes a few of the cell structures. So for example, in the top image in the middle where it says most plant and animal cells, that's a light microscope image showing you a plant cell. And you can see some of the internal structures. For example, those knotted structures are chromosomes. However, to see most of the structures inside of a cell, you need something called an electron microscope. And electron microscopes can show you small details, even so far as to show you some of the atomic structure of some of these molecules, but we still can't see at the level of atoms quite yet. So, you have two different major types of microscopes that we use to study cells. Light microscopes have a limitation in that they can only magnify about 1,000 times effectively, which means we can't see some of the smaller structures, so we can't see a lot of organelles. We also can't see viruses using light microscopes. But they do have an advantage in that we are able to see living cells. So at some point we will look at some living cells under a microscope. And be able to actually see some movement, and you'll also see some of those images on some of the videos that I link to in the comments or in the description of this video. To see cellular structures, you need to use something called an electron microscope. So light microscopes reflect light off of an object and use that to allow you to see an image. Electron microscopes, though, focus beams of electrons instead. That's where they get their name from. They're also not an object you can look at. You're not looking through a lens and seeing an image. You will need some more fancy equipment for that. Some of it might use some computers to help reconstruct the image. Some of it might be a little simpler than that. But anyway, there are a couple of different types of electron microscopes. Scanning electron microscopes tend to look at the surface of something. Transmission electron microscopes are able to actually see inside of a specimen, so they can see inside of a cell. But I will tell you there is a limitation. The preparation process for preparing specimens to be seen with an electron microscope does kill them. So you can't see any living structures using these. So this is just showing you the electron microscope is on the bottom left, the light microscope is on the top left, and on the right side you see two different images produced by an electron microscope. The top one is showing you the surface of a cell, so you're able to see the outside of cilia, and that's a scanning electron microscope, whereas the transmission electron microscope allows you to see internal structures, so you can actually see inside of those cilia. There's some other tools that are used to study cell parts as well. This is not in your notes, but I just wanted to show it to you. So what you can do is something called cell fractionation. You basically take a cell, you either treat it with a chemical that bursts or lyses the cell membranes, or you can, like in here, they're just showing you sticking it in a blender. But some agitation to the cells can break them open as well. Then you put them in an ultracentrifuge. So you may have seen when we were doing our PCR lab that we had a microcentrifuge that spins objects in order to either concentrate at the bottom of a tube, or in the case of this ultracentrifuge, you can actually use it to separate out parts of cells. So it spins super fast, and when it does, it's going to separate out substances based on their mass. So the heavier things are going to go to the ends of the tube, and the lighter things will be farther from the ends of the tube. What you see on the bottom right of the diagram is that these cells have been separated into some distinct layers which have different organelles inside of them. Then you take them, you can view them through an electron microscope, and you can see these structures in more detail. So I mentioned what all cells must do functionally. They have to maintain homeostasis, etc., etc. Because of that, every single cell on the planet has these structures. So the DNA has to pass on its traits to its daughter cells or daughter organisms. Cell membranes are necessary to separate a cell from its surrounding environment, help it maintain homeostasis. Ribosomes are going to produce proteins, and all cells produce proteins. And then the last two things are structural components. All cells have cytoskeletons, just like most organisms have some sort of skeleton of a sort, even if it's an endoskeleton or an exoskeleton. I'm talking about animals, by the way. And so cells have skeletons as well. They're just made up of proteins and small molecules as opposed to bones or some sort of outer crunchy shell. Cytoplasm, including cytosol, is also a part of every cell, and we'll talk about that in a moment. So before we move on to talk about the parts of cells, let's review the different types of cells. There are prokaryotic cells and eukaryotic cells. And by the way, this is not on your notes either, but there is a Venn diagram on your notes, which the next couple of slides will help you fill in. So notice that we have one major type of organism that is a prokaryote. Those are bacteria. There are a couple of different types of bacteria, but they are all prokaryotic. In fact, you can use those two terms synonymously. Eukaryotes, on the other hand, are broken down into three major categories. There's actually a fourth one, but we're not going to worry about it. So they're animals, plants, and then fungi. Fungi we're not going to talk about in very much detail. We'll focus a little more on animal and plant cells. So, prokaryotes are non-nucleated. They tend to be much smaller, and they don't tend to have a lot of complex internal parts. So they don't tend to have a lot of membrane-bound organelles. In fact, that's part of the definition of a prokaryote. They have no membrane-bound organelles. They do have DNA. It's not separated from the rest of the cell. Instead, it's concentrated in one region of the cell called the nucleoid. And the two major groups of prokaryotic organisms on the planet are bacteria and archaebacteria. They are not really easy to distinguish unless you start to look at either DNA or you start to look at like some of the chemical components of them. Eukaryotes, on the other hand, do have membrane-bound organelles. The biggest, most defining structure of them is they do have their DNA separated from the rest of the cell by a nucleus. And a lot of the other organisms, all of the multicellular organisms that we can see with the naked eye, are eukaryotes. So those are protists, fungi, animals, and plants. So, very quickly, prokaryotic cells are all enclosed by not only a cell membrane, but also a cell wall. There are these molecules called peptidoglycans. They're a combo of protein as well as carbohydrate, and they make up the cell walls of bacteria. Then you can also have some other layers outside of the cell wall. So some bacteria actually have an inner membrane and an outer membrane that's separated by a cell wall. And then still other bacteria have this kind of slimy layer of polysaccharides, which are carbohydrates that surround them. And it can be protective, but it's also useful because we can use it to try and identify species of bacteria, especially if we're trying to see who's being infected by what. You don't need to know a whole ton of detail about, by the way, all of those structures surrounding a bacterial cell. And on this diagram, you'll tend to see the most important parts of a bacterial cell. What's important to note is all the ribosomes are free floating inside the cytoplasm. You don't have a separation between the nucleus, between the DNA and the rest of the cell. It'll become a little more important when we talk about DNA in more detail next 6 weeks. You can have some more complex functions in a prokaryotic cell. For example, there are prokaryotic photosynthetic creatures. Most notably, cyanobacteria are kind of the most typical type of photosynthesizing prokaryote. They are bacteria. They don't have internal membrane-bound organelles, so they don't have chloroplasts. But what they do have are these infoldings of the cell membrane that kind of form this internal membrane system, and that is what allows them to perform photosynthesis, which we'll talk about in more detail when I talk about photosynthesis more towards the end of the semester. Eukaryotic cells are able to become not only much larger but also much more complex because of their membrane-bound organelles. A big example is the fact that you have these organelles called lysosomes, which are digestive organelles, and so they'll have a much lower, more acidic pH, which aids them in digestion, but you wouldn't want that exposed to the rest of your cell, because if so, it would start to eat away the rest of your cell components, and that would be like, letting your digestive juices leak out of your stomach. Not healthy for a human body, just like those digestive enzymes in a lysosome need to be sequestered away from the rest of the cell. So here you see not only a diagram of an animal cell, but you also see electron microscope images of the various parts that have been isolated for you, so you can see like a nucleus, you can see elements of the cytoskeleton, etc. I will let you look at this at your leisure and instead move on to some more of the organelles that were not visible on the previous diagram, but this time they're showing you the organelles in a plant cell. So you can see a couple of different views, a couple of different images. And now let's look at organelles that all cells contain. All cells have cytoplasm. So this diagram, I know it looks like some fancy random abstract pattern, but it's actually a representation of the cytoplasm and all of the proteins that are stuck in it. So cytoplasm is everything outside of a nucleus, and we tend to see it in diagrams as mostly empty space filled with a few organelles, but in reality it's chock full of proteins and it's chock full of organelles. It does also contain some fluid called the cytosol, which does have some important chemical reactions happen in it, and it's also the fluid in which all of these proteins, all of these organelles are floating around. The cytoskeleton is something that's also found in all cells on the planet. It helps support cells, it helps them maintain shape. It can anchor organelles in place, or it can help move them around. There are also these special classes of proteins called motor proteins that, in conjunction with the cytoskeleton, are able to help move either parts of the cell or move the entire cell itself. There's also something called cytoplasmic streaming, which is the fact that within cells, organelles are moving around, and that is due to these motor proteins as well as the cytoskeleton allowing this movement. And it can also anchor basically to the cell membrane on one side, and then allow the cell on the other side to anchor to stuff outside of the cell or extracellular structures, so that way the cell can stay in place. There are three major components that make up the cytoskeleton. In order of increasing size, you have microfilaments, intermediate filaments, and microtubules. You can get a little bit of a hint of their relative size based on their names. The microfilaments are the tiniest ones, intermediate are kind of in between. So microfilaments and microtubules have this important property called dynamic instability. What happens is they're made up of all of these little subunits, and they can be very quickly broken down or built back up. So microtubules and microfilaments are able to be broken down, built up very quickly inside of the cell in order to change the cytoskeleton or to even move parts of the cell. Most of these fibers are directional, meaning that they have an end to which you can add subunits and another end where you can remove subunits, but you can't like add subunits to both ends. So some details about each of these types of filaments. Microfilaments are made of something called actin. Their specific function is they are going to resist pulling within a cell, and they can help move cell parts around. They're involved in the contraction of muscles. They're also involved in cytoplasmic streaming. You also have them make up like structural parts of specific cells. So here we have microvilli, which are these little finger-like projections sticking up from your intestinal cells, and you can see that the core of them is made up of these microfilaments that kind of can help keep those microvilli in the proper shape. I'm going to include links to both of these videos that the blue hyperlinks are indicating. They'll show you both amoeboid movement, which is a movement of these single-celled creatures called amoeba that are able to kind of move around and extend what are called these pseudopods, which literally means kind of feet, that allow them to move. They also allow them to engulf smaller creatures and eat them. And there's another video which you'll be able to see in the links in the description that will show you cytoplasmic streaming, so it'll actually show you organelles being moved around inside of a cell. Intermediate filaments are made up of proteins that are found in the keratin family. Keratin is the same protein that makes up your hair and nails. Obviously, you don't have like hairs running through your cells, but you have these much smaller, finer, thinner units. They don't tend to be built up and broken down, so your intermediate filaments are going to be a relatively permanent, stable part of the cytoskeleton. That's used to anchor parts of the cell and also helps maintain the shape of the cell. Microtubules are made up of these proteins called tubulin. Don't need to know much more about tubulin. Microtubules also are shaped like these hollow tubes. We'll see a couple of other diagrams that'll let you see that structure a little better. They are very important because motor proteins will use them to help either move stuff around or to help move parts of the cell. They're also important because they're used to move chromosomes around during mitosis. And they are essential for making up cilia and flagella, which we'll talk about on another slide. So the source of organelles the source of microtubules inside of a cell is mostly these structures called the centrosome. It is actually called something different inside of a plant cell. It's called the microtubule organizing center, or the MTOC, which we'll see when we talk about mitosis in plant cells. Each animal cell has a pair of these centrioles, which are made up of these kind of sets of microtubules arranged in a ring, and they are really important in mitosis. Microtubules are also important because they form the internal parts of cilia and flagella, and it's the presence of these microtubules that are linked by motor proteins that actually allow these cilia and flagella to move back and forth, which is what allows single-celled creatures to move around. That's what that really fuzzy looking structure in part A is. That's a single-celled organism that's surrounded by cilia. So this is not in your notes, it's just extra information. You have multiple motor proteins. My favorite protein in the entire body, by the way, is this guy kinesin. One of the animations that I'll link to in the description actually will show you kinesin and why I think it's so cool. It literally walks along a microtubule to move stuff around. It's pretty darn cool. All right, let's get to our next organelle, ribosomes. Ribosomes are these little tiny organelles. They're the smallest organelle. They're also just a very large molecule, so they're kind of that border between, they're on the borderline between a molecule and an organelle. They're made up of ribosomal RNA, which is bound to proteins, and they have a large subunit and a small subunit. Apologies. Like I said, the noise of cats playing. Their function is to translate RNA. So they'll take the genetic code in RNA and use it to create a protein. So they are the sites of protein synthesis. We'll talk about them in much more detail when we talk about DNA next 6 weeks. In prokaryotes, they're just floating out in the cytoplasm. In eukaryotes, they can either be free-floating or they can be attached to rough endoplasmic reticulum, which we'll talk about in more detail when we talk about the ER. In the cytoplasm, they can form these structures called polyribosomes, where you have a bunch of them attached to a single RNA strand, and I mostly include that because it'll be slightly important when we talk about the regulation of producing proteins, but they also just kind of look cool. So here you see some ribosomes. Some of them are attached to ER, some of them are free-floating, and we're about to get into some of the membranes and stuff inside of a eukaryotic cell, which, yeah, sorry, I did the order of that wrong. So let's talk about organelles found only in eukaryotes. Well, in eukaryotes, you have a nucleus, which helps define a couple of terms. You have this, that you can divide up the internal parts of a cell into two major structures, the cytoplasm and the nucleoplasm. The nucleoplasm is all of the stuff inside the nucleus. The cytoplasm is everything inside of a cell except for the nucleus and the nuclear membrane. So it's basically everything in between the nuclear membrane and the cell membrane is cytoplasm. By the way, we're going on to organelles that are only found in eukaryotes at this point. There is this large system of structures called the endomembrane system, and you may not have heard of it before, but you've got a lot of familiar structures in it. These basically are a lot of, their major function is protein synthesis, and then getting those proteins ready for whatever function they're going to perform in the cell, or even outside of the cell. So technically the nucleus is part of the endomembrane system, as is the endoplasmic reticulum, Golgi, lysosomes, vacuoles, and then the cell membrane is part of this as well. These structures are either physically interconnected or they can be connected by the movement of these small balls of membrane called vesicles. So this is the pathway through which proteins are going to move. So they are going to start as genetic information in the nucleus. Then that information is going to be transmitted via RNA to a ribosome. A lot of ribosomes are attached to the ER, and so ribosomes that are attached to the ER will produce proteins that will either get released into the ER to be like further modified, or that will become part of the membrane itself. So membrane proteins are produced in the ER, and then these guys are usually exported out to the Golgi, which will then modify them in preparation to release them from the cell. So here we see this in diagram form. You can see that the nuclear membrane is actually physically interconnected with the ER. Then stuff can be moved via vesicles, so those small balls of membrane, to the Golgi. They can go from one sac of the Golgi to the next sac until finally they are going to almost melt into, or they will fuse with the cell membrane and then release their contents out of the cell. I am going to include the links to these animations in the description, but I highly recommend watching them, especially the evolution of the endomembrane system, because the nucleus and the endoplasmic reticulum have some weirdness to them. Like, you may wonder, why does the nucleus have two membranes instead of just one membrane? That's actually answered in that evolution of the endomembrane system animation. We'll also try and go through them in class too. So we've mentioned the nucleus a couple of times. Let's talk about it. Its job is to store DNA. It's also where an RNA is transcribed, and it's also where ribosomes are produced. So ribosomes are created in this dark structure in the center of the nucleus called the nucleolus, also sometimes pronounced nucleolus. Just depends on it. I'll pronounce it either way. It's just random. So, like I said, there is a nucleolus, which is usually a darker spot inside of the nucleus. In diagrams, you'll usually see what looks like a very complicated folded ribbon, which is the DNA. There are all of these nuclear pores, which are holes in the nucleus, but they're not just like holes that anything can pass through. They are actually surrounded by these nuclear pore complexes, which are proteins that are designed to help regulate what goes into and out of the nucleus. The nucleus stores the instructions for not only what proteins to make, but when to make them and regulation of them. It's very important that there's communication between the nucleus and the rest of the cell, but it's also important that you make sure foreign DNA, like from bacteria or viruses, does not get into the nucleus. And the nuclear membrane is continuous, physically interconnected with the endoplasmic reticulum. So here we see some electron microscope images of various parts of the nucleus, as well as diagrams. I just like to make sure you guys have plenty of diagrams to look at so you can visualize what these look like. Now, let's talk about the structure that is interconnected with the nucleus. Let's talk about the ER, or endoplasmic reticulum. Endoplasmic reticulum is a lot of the membrane that's found inside of a cell. Depending on what the function of that cell is, you can have a ton of endoplasmic reticulum, or you can have just enough to keep the cell functioning. The inside of the endoplasmic reticulum is called the lumen, also sometimes called the cisternal space. I'll typically refer to it as the lumen. And you have two types of ER. We'll talk about each of those right here. So they have different functions. Rough ER is what a lot of people tend to picture when they think of ER, because it's the stuff that's studded with ribosomes. So since it's got ribosomes attached to it, and ribosomes are involved with protein synthesis, hopefully that'll help you remember. Rough ER is all about making proteins and modifying them. So they can be modified where you can add carbohydrate groups onto them, turn them into glycoproteins, you can add other chemical groups, and you can also tag them for being delivered to a specific part of the cell. Any protein that's going to be released from the cell or secreted, as well as most of the proteins that are going to be sticking out of the cell membrane, will end up either passing through or being produced in the rough ER. This is also a spot where you produce membrane. So any membrane that's going out to the cell membrane tends to be produced in the rough ER. Smooth ER, on the other hand, generally works more with lipids. It does not have any ribosomes attached to it, and it also looks a little different, which we'll talk about on one of the next slides when we look at some pictures. So it also detoxes, so it can detoxify harmful chemicals. It's involved in breaking down glycogen, which is the main storage starch in animals, and it's involved in metabolism of lipids, making lipids, modifying lipids, etc. It can also be used to store calcium, and also for the metabolism of both carbohydrates and steroids. Steroids are a type of lipid, which we'll talk about more when we do biochemistry later. So the structure of these two is different as well. Rough ER tends to be these like flattened folds of membrane, and smooth ER tends to be a bunch of tubes. So if you look at this diagram here, you can see the difference in that not only does rough ER have all of those ribosomes attached to it, but it looks like a bunch of messy pancakes, whereas smooth ER instead looks like kind of a weird tubey network. So you may have noticed that I'll have Golgi and then I'll have three names after it, because I've seen the Golgi, the organelle referred to as the Golgi, referred to as the Golgi apparatus, Golgi body, Golgi complex. It just kind of depends on what text you're reading. So I just wanted you to be familiar with the fact that all of these terms are used. Sometimes I'll call them the Golgi ABCs just because it's got all those terms, but most of the time I'll either just call it the Golgi or I'll call it Golgi body. So what it does is it receives things from the ER, usually vesicles kind of cart that in, and it's going to sort molecules for different destinations. It can concentrate them, so build up a higher concentration, and package them so that they can be sent out into transport vesicles to go to either different locations in the cell or also be sent to the plasma membrane so that way they can be exported from the cell or secreted from the cell. So it's a system of these flattened sacs called cisternae and vesicles that melt into or out of the cisternae. I think of it like a super messy stack of pancakes, even more so than I think of the ER like that. You have two distinct sides. The cis region is going to face the ER, and the trans region is going to face the cell membrane. If you think of it, the word trans means across. So the trans face is going to be sending stuff outside of the cell and across any barriers between cells. And then in between those two regions, you have the medial region. You're usually going to have vesicles coming into the cis face and leaving from the trans face. That's just showing you more images. Let's move on to lysosomes. Lysosomes are these digestive, they're basically little stomachs almost for a cell. So there's these little balls of digestive juices. They're important for unicellular organisms because they're necessary to digest their food when they take it in, but they're also important in multicellular creatures like us. They're used to recycle damaged cell parts. So if you have an organelle that's messed up, you'll probably encase it in a vesicle, a ball of membrane, join a lysosome to that. The lysosome will break it down, and then your cell can reuse any of the molecules that were used to make up that organelle and use them somewhere else. They are also important when you have apoptosis. So apoptosis, remember, is programmed cell death, and if you have a cell that's undergoing apoptosis, it might just release the contents of its lysosome inside of the cell and destroy everything. So lysosomes are involved in digestion. So what you're seeing in this diagram is you're seeing a unicellular creature taking in, by endocytosis, some sort of food particle that gets joined to a lysosome, which ends up coming out of the Golgi. The lysosome then binds to or fuses with that vesicle containing the food, digests it, any of the nutrients important molecules are absorbed into the cell, and then that kind of waste containing sac can then be fused to the cell membrane, exocytose, and spit out any of the trash components. Here you're seeing the same thing happen, except on B you're seeing a cell that's digesting some of its messed up organelles or old organelles, and then is able to recycle its components. Mitochondria, I know you guys think of them as the powerhouse of the cell, but you're going to need to modify your internal definition of them and start referring to them as the organelles that perform cell respiration or the organelles involved in producing ATP. And in general, if you have a cell that needs to use a lot of energy, it's going to have a lot more mitochondria. They are a double membraned structure. They have two membranes, an outer membrane and an inner membrane. The inner membrane is highly folded, and then in there it's got a matrix that contains not only enzymes, but it also contains separate DNA from the rest of the cell. When we talked about evolution, we talked about endosymbiotic theory and support for the idea that mitochondria were once separate, independent bacteria that then got endocytosed or taken in by ancestor of modern day eukaryotes and ended up forming this nice, happy, symbiotic relationship. That's where the separate DNA comes from, and it also has some ribosomes of its own. So now we're going to look at a class of organelles that are mostly found in plants. So these are called plastids. They are double membraned. They do have their own DNA, and they're probably the result of endosymbiosis. A lot of times they'll store materials, but they can also become other types of organelles. And part of the reason why we're talking about them is the fact that chloroplasts are a type of plastid. So I have some details about a couple of other types of plastids that are not on your notes. Didn't think they were that important, but I just wanted to show you some examples. So chromoplasts are these plastids that have different pigments. They're really important for the parts of a plant where color is an important signal, like flowers and fruits. Leucoplasts are going to store molecules. So in the roots of a plant where you're storing starch, you're going to have a lot of leucoplasts. And then let's talk chloroplasts. They perform photosynthesis. They are fairly complex. They have sets of membranes inside of them called thylakoids. These thylakoid membranes contain the proteins and enzymes that are responsible for photosynthesis. And they do also have their own ribosomes. They also have their own DNA, etc. We'll talk about the structure of chloroplasts and the function of them in more detail when we talk about photosynthesis. Vacuoles are also, they can have multiple functions. When you guys were doing your organelles chart, a lot of you guys were talking about the central vacuole. That's a very specialized vacuole that's found in plant cells, but you can have vacuoles in other cells as well. In a plant cell, a central vacuole is a storage space for water and ions, but it's also important for storing water that can help create the turgor pressure that pushes out against a cell wall. You can have other vacuoles that are in other parts of a plant that contain pigments. You can have some in seeds that contain digestive enzymes. And then protists also have an important vacuole called a contractile vacuole. Basically, if they're in fresh water, they're always sitting in a hypotonic solution. So water is always entering the cell. In order to not explode or lyse, they have these contractile vacuoles that will fill with the water that's entering the cell, and then they'll expel it, squeeze it out periodically to make sure that they don't lyse. All right. So here is an image of a central vacuole in a plant. Yes, it takes up a large part of the plant cell's volume. Now let's talk about the stuff outside of the cell, because these are equally important. They're usually secreted, released from the cell. So a cell will build its own extracellular environment in a lot of cases. In most eukaryotes, you're going to have two things. You're going to have some fibrous molecules that are going to create a lot of the structure of the extracellular parts, and then you'll have this kind of gel that the fibers are embedded in. Depending on which fibers you use and what's in the gel-like matrix, you're going to produce different structures. So the most important extracellular structure is a cell wall. So let's look at plant cell walls. They have a number of functions. They're a barrier to entry into the cell, so a little bit of homeostasis. They also provide support. Remember, plants don't have bones, so a lot of their physical structure that supports them is cell walls. Unless you're talking about trees, they have other things. And then they're also important during growth and development. What ends up happening is a plant cell will excrete this fibrous substance called cellulose. It's the stuff that makes plants crunchy. If you ever eat celery, celery has a ton of cellulose, which is why it's really fibrous and kind of hard to chew. Then you have a bunch of polysaccharides, which are carbohydrates, and proteins that are kind of released out into the extracellular space as well, and then that forms the cell wall. You can have a couple of layers of cell wall, depends on the individual plant you're looking at. Animal cells also have this extracellular structures. It's called the extracellular matrix for us. Sometimes it's abbreviated the ECM. For animals, its main function is to help hold cells together into tissues. So especially in the type of tissue called connective tissue, which helps hold stuff together, this extracellular matrix is really important. It can also be used for some other things, like for example in kidneys, you can use it to filter, and it's really important to help cells move around. So during development, when we were all fetuses inside the uterus, a lot of cells were moving around all over the body, and the way that they were able to get to the proper place, so for example, to form parts of our nervous system. They were orienting based on the extracellular matrix. So in animals, it tends to be mostly the fibrous component is collagen. Then you have these proteoglycans, which are basically proteins linked with carbohydrates that are going to help link all of that together. So you see a couple of examples of extracellular matrix here. You see those fibrous components as well as some of the other components like the proteins that are help link and cross-link these different fibers. Here's just another image showing you like a proteoglycan and its structure, showing you how the extracellular matrix is anchored to, like for example, here we have fibronectin, which is a protein that is bound to an integral membrane protein that then binds to the extracellular matrix, helps hold everything in place. You have some other molecules that are also used to attach to the extracellular matrix as well. Integrin, in one of the videos that I'll have a link to in the description, it's going to show you integrin, and it's going to show that integrin allowing a white blood cell to roll along the inside of a blood vessel. Also, you can have in animal cells and plant cells these junctions or these spots where cells are binding to each other that will let them not only stick to each other, they can also allow communication or even direct physical contact between the cytoplasm of two cells. So one of these that allows direct physical contact are plasmodesmata. Plants don't have a heart. They do have a circulatory system. In essence, their vascular system, but there are also these interconnections in between plant cells that almost tunnel through cell walls. They're called plasmodesmata. They do contain cytoplasm, so materials can move directly from one plant cell to another. For example, if one plant cell ends up getting infected, it can produce signaling molecules to neighboring plant cells that say, hey, there's an infection, increase your defenses. Animal cell junctions tend to fall into three categories. You have tight junctions, which are basically used to kind of almost super glue different cells together. So any place you want to have a watertight barrier or a very tight barrier, so for example in your stomach, you're going to have tight junctions binding all of your cells. You don't want the contents of your stomach leaking out. Desmosomes do link cells together, but they are not as tight junctions, so they'll allow stuff to move in between cells. And then gap junctions are pretty rare in mature adult animals, especially like adult humans. We don't have a lot of gap junctions in us. They allow movement directly between cells, so there's continuous cytoplasm in between those. We don't tend to have a ton of gap junctions. It's a bit of a weakness in the barrier that surrounds your cell, because if one cell is infected, rather than having to go through the cell membrane of a neighboring cell, whatever infectious agent can just go through a gap junction. So you don't want to have a ton of those. Here's more details and more detailed diagrams that you guys are free to look at. And here are some electron microscope images. So that is the end of our lesson on organelles. I know it was a long one, but I did try and shorten it some.